So anyone now who logs in can join. So it's being recorded onto, it's being recorded onto, it should be anyway, onto this computer. Yeah, so you can set it within, it depends what the settings are in.
So Rebecca, we've just got it sorted out. You'll have to front lights or something. So one thing we'll have to
You could
do
put it on
here.
mute if you wanted to while we're.
That one? No.
Yeah.
Mute. No, just turn it on. Just stand next to the other. Yes. Okay, good afternoon, everyone. I think we'll wait one more minute so a few more people can join online. So there's quite a few more registered than are popped on, and then we'll start in maybe a couple of minutes. Sounds good. Sounds good. And it is recording according to the cloud. Yep. Yeah, all good. Yep. Participants. We've got about 40 minutes, is it? And then some questions. Yeah. Yeah. I'll keep a bit of an eye on the time, but should be okay. Yep. Okay. Good afternoon, everyone. It's great to have these seminars back in person. We've in fact got a hybrid today. We've got an audience sitting in front of us down in the McLeod Theatre, down in the Charles Falker building, and also quite a number online. And this is also being recorded, which we'll share later. So for those online, if during the talk you want to populate with question and answers, please do so. Use that rather than the chat function, but we'll see how we go. And Luke's got a few questions that he's going to ask the audience, so please interact with that online. So it's my great pleasure to introduce Associate Professor Luke Mosley, who's been with the School of Ag, Food and Wine for since the beginning of the year, but prior to that was at the university probably for the last 7 years. Yeah. Situated in biological sciences, but often based here at the Waite anyway. So it's been a member of that community here for quite a number of years. And prior to that, he had 10 years with the Environmental Protection Agency based in the Murraylands. Is that right? Yeah, mainly working on the River Murray and things, yeah. So Luke, as many of you know, and I'm sure you recognise, hails from across the Tasman with his degree at the University of Otago and PhD as well, prior to coming to South Australia, and has been among his other roles, has had a lot of professional community interactions with the Soil Association of Australia, and was president of the Soil Science Australia from 2018 to 2020. So there's done some great community roles there as well as some stellar soil science, and also in spare time, I can attest to this, he's an excellent coach of school sports. I've learned a lot from him, especially tolerance. I'm still working. The Gilliam child was one of the best behaved to know. But real pleasure to have Luke to tell us about his science today, particularly some work that he's performing, and I think we'll talk about some of the GREC work with soil and soil pH, so acidic soils. So we're very much looking to hear for that. So
Thank you, Matt, for a very kind introduction, and thank you all for attending today in person and online. And yeah, today I wanted to, I guess, pose a bit of a, I guess, a question to start with. How well do we actually understand soil pH? Soil pH, as I'll talk about in my talk, if I can get the slides working. Here we go. It has a fairly long history, which I'll outline briefly some of the history of measuring soil pH. Firstly, I'm going to talk about pH in general. How well do we actually understand pH as a variable? And I'll ask you a few questions, and we'll challenge our knowledge there. We'll talk briefly. pH is talked about as a master variable for soils because it can control and influence a range of other biogeochemical processes in soils. So we'll talk about that briefly. We'll go on to talk about some challenges, or as I see them, with soil pH measurement, and perhaps some issues with how soil pH measurements are being done in various certain contexts. And then I'll talk to you a little bit more, and this coming into the work. I'm currently working on a GRDC project with PIRSA, and Brian Hughes is leading that on soil acidity in South Australia. And I think we're getting a much better handle now on how variable pH is in the landscape through that project and other projects. So I'll show you a little bit of that, and also touch on some new measurement techniques that my students and others have been working on here at Waite for the last several years. So we'll start off with a question. Who can tell me who invented pH? Does anyone in the audience got an answer there? No? Yep. Someone, all right. Someone who did the equation to measure pH, yep. So pH was invented by, defined first by Sorensen in 1909. So yeah, he's there with one of his primitive measuring devices. And so with, pH itself has been around as a variable for a little over 100 years or so. Next question. And I'll mention he got nominated for the Nobel Prize. He won a number of awards for his work on pH and some other chemistry of solutions he was working on. He never won the Nobel Prize, but he was honoured with a Google banner. Also, as you can see there. Next question I'll post here: did beer help in the invention of pH? It probably gives you a little bit of a clue there with the bottle there. So, an interesting fact. So, Sorensen invented pH while running experiments with beer. At the Carlsberg Research Lab in Copenhagen. So he was director of the Carlsberg Lab at age 33. So I think a fairly young age for a major director role at something as important as the Carlsberg beer. So he was obviously a very talented young man, and he was primarily studying fermentation, and in particular the formation of amino acids, which many of you know, the building blocks of proteins. So he studied the effect of hydrogen ion concentrations on those proteins, and because they were, you could see that how important they were, he introduced the pH scale as a simple way of expressing it. So can anyone tell me what does the P in pH stand for? Check the Q&A. No, all right. That's all right. I'm not sure many people can understand that. There was, and I'll point to get the next slide working. So this little excerpt here is, I'll just move this video for a second. is excerpt from Sorensen's original publication on it, and I'm not French, so I'm not going to read that, but there was a paper done by Jens Norby in 2000 when his father actually worked with Sorensen, so he had a connection to the Carlsberg laboratory, and he was concerned that there were a lot of false associations, which he puts in a table here in the paper on what the meaning of P and pH is, and there some of these false associations that you hear of, according to him, he's done the most detailed analysis, I think, of it, is that people talk about power, they talk about a potential of hydrogen, all these different factors. So there's no clear evidence that that is what Sorensen meant, and Jens Norby concluded that it seemed that the little p in pH originates simply as a consequences of Sorensen's arbitrary choice of the letters P's and Q's. So you say, mind your P's and Q's. It's interesting. So for the two solutions, so by choosing solution Q to have the H plus concentration 1, which I read as a calibration solution, the unknown concentration of H plus gets the concentration symbol CP, or 10 to the negative P, and then he defines that as pH. So we're traveling fairly well in our misunderstanding journey so far with pH itself. The next question I'll ask you, does H plus, so we're talking about H plus ions as a measure of pH, do they even exist in solution? Any comments from the audience? No. I know Mike McLaughlin's online, but he'll probably comment. But in fact, when people have attempted to measure protons in solution, there's an incredibly low concentration. I think they estimate 10 to the negative 130 is the actual proton concentration. And the reason for that is water's got a very strong affinity for hydrogen ions. So in solution, you're actually talking about hydronium ions, where the proton here has attached to a water molecule. And this is a statement from the American Chemical Society that when you see an aqueous chemical equation with a symbol H plus, be assured that the actual ionic species is H3O plus, the hydronium ion. These are just some interesting facts. And then how do we define pH? Well, this is another sort of interesting historical journey with pH. The original definition of Sorensen was pH is the negative log of the hydrogen ion concentration. It's subsequently been defined by the International Union for Chemistry as the negative log of the hydrogen ion activity. So the definition itself has morphed somewhat from Sorensen's original, although people still use concentration in some contexts. And these two are not equivalent. They only converge in very dilute solutions. So essentially ionic strength approaching 0. So not really in many soils you'd consider that would be the case. So you've got to be careful, I guess, how you define pH as well, particularly in more saline solutions. And then the next question I'll ask you, if we define it, say, as if we adopt the International Union of Chemistry definition of hydrogen ion activity, can we actually measure hydrogen ion activity? Does anyone want to guess? All right. So activity is concentration times the activity coefficient. So it's basically what you find when you've got a very dilute solution, they call the activity or the potential of that ion is the same as concentration. When you've got other ions in solution, so when the salt salinity ionic strength increases, those interactions lead to that primary ion having less activity per se, or less ability to interact in reactions because it's interacting with some of the other ions in solution. So back to the question, and this is from that IUPAC document there, the definition of pH scales. It's actually immeasurable. So we don't actually measure the hydrogen ion activity directly. which is another interesting point. So it's really an operationally defined definition where we calibrate on solutions, standard reference solutions, which are mentioned down here. Many of you would have used those buffer solutions to calibrate electrodes, but you are calibrating to an operational definition. You are not measuring the single H+ ion activity. So that is a bit of a background and some of the perhaps more interesting misunderstandings to do with pH that have a little bit of interest for, more at an academic level, but they do flow into some measurement aspects. So now we'll move on to pH as a master variable in soil. So many of you soil scientists I can see out here have worked with pH in soils, and when I talk about a master variable, as I mentioned at the start, we're talking about how that variable can control and influence the concentration or behavior of other elements. And probably one of the most well-known factors in soil influences is on nutrient availability. And you can see here a range of different nutrients, nitrogen, phosphorus, potassium, etc. And you can see as the pH decreases, so you're going into acid zone, the availability of many key nutrients decreases. But that's not all the case for all the elements. There's some that don't decrease at low concentration, in fact increase, things like iron and manganese, and some that increase at high pH like molybdenum. So it's really dependent on the element and its chemistry and how it interacts in the soil. But overall, pH, very strong control on nutrient levels in soil. It's also got a very strong control on microbial activity. This plot shows the basal or the microbial respiration for two different soils as a function of pH. So you're getting down here to a 3 or 4. So you can see that influence of pH on the microbes. And that could be also due, I think, to some metal toxicity issues as well as H+ issues as well. But you can see, big decrease in microbial activity, also decrease in microbial biomass and microbial carbon and nitrogen. Metals have a really strong relationship with pH. Aluminium becomes available below pH around 5.5 in water or below 5, say in calcium chloride. You can see here some of the effects of aluminium present and low pH conditions where the roots for the high aluminium plants really poorly developed, and subsequent effects on yield, as opposed to a more slightly higher pH, but probably still below optimal, but lower aluminium levels there. And you can see in quite narrow bands that these metals change. So this is the mole percent bound. So if we look at some of these metals here, say copper, between about 5 and 5.5, it goes from 0% bound to the soil particles. This is ferrihydrite, which is an iron oxide in this particular experiment, up to over 80% bound. So within a half a unit pH, you can get very large changes in metals depending on where you sit on these particular metal sort of isotherms. And it's similar for some of the anions in reverse. As the pH increases, there's changes there as well. CO or pH is integrated or integral to the carbon dioxide system, both atmosphere, oceans, soil. And soil pH has a role, I guess it interacts with carbonate ions. It can form carbonic acid or H2CO3 bicarbonate. So that's got HCO3, so that's got the proton attached, or dissociate fully to just carbonate ions. So it's really integral. And there's evidence, I guess, emerging, and we've known for a long time that CO2 in soils affects pH, and microbial activity can alter pH through building up CO2 levels. But there's evidence now that atmospheric CO2 is also interacting with soils and changing the pH, just as it's changing the ocean pH as well, and interacting with the carbonate system in the ocean. So really an incredibly important parameter for soils. So if we move on now to how do we measure pH in soil and go to some of the, firstly, the history of that. Well, from what I can ascertain, some of the earliest soil pH measurements were made in the 1910s by a series of researchers, Fisher, Sharp, and Hoagland and Gillespie. There's 3 early papers there from 1914 to 1916. And this, was probably only 5 or so years after Sorensen. So that's quite interesting to me that the soil scientists adopted very quickly pH measurements after they were defined by Sorensen. And this just shows you an example out of interest from this Sharp and Hoagland paper, and says for the hydrogen electrode work or the pH measurement, a suitable quantity was found to be 2 cc's, which is mils per gram of dry soil. So that's a soil to water ratio of 1 to 2. And they also used indicators for that work. They also air dried the soil and they did some coarse sieving and grinding. And that's essentially the pH method. You can argue about the soil water solution ratio, which I'll talk about, but it's essentially the method that's used today for a large, to a large extent. That what they adopted back then is variations of a theme, but largely what they're doing today. And you can see here how they express it. The hydrogen ion concentration expressed as the same as Sorensen. So they've adopted Sorensen's scale, the early soil scientists, and really using it. So I think that acknowledged that soil acidity or impacts of soil acidity on agriculture by that point, but they really hadn't, I think, had really good measurement techniques up until that point to really be able to define it, measure it accurately. So I mentioned that 1 to 2 ratio. Well, that's an area that's been researched for a long time, and some of those early experiments varied ratios from 1 to 1 right up to 1 to 250, and it's I guess soil scientists tend to have settled on the 1 to 5 ratio now. And I'll talk a little bit more about that, but there is a strong influence that people don't realise on soil water ratio on pH, and we need to be careful how we treat data and how we use soil water ratios to infer field pH conditions. So this just shows you a plot of 2 soils, and you've got pH here, and this is the solution salinity, and this is different soil water ratios. So from 1 to 5, so say 10 grams of soil and 50 mils of water, coming up to where you've got slightly more soil than water, and then a saturated paste extract there. And these other ones are calcium chloride extracts. But you can see as you're transitioning down from that 1 to 5 ratio, which is now the common, I think most widely used, less than commercial laboratory ratio, you can see coming down to a saturated paste, which would generally think would be most representative of field conditions in terms of soil water ratios, then of saturated soil water ratios. Then you're down at, you've dropped over a pH unit in this particular soil, and slightly less in this soil, maybe 0.7 or 0.8, just by varying the soil water ratio. So I guess if you're going to then infer that the pH of the soil in your agricultural system was this, then maybe in the field it's this. So it's an interesting area that I think we neglect a little bit too much as soil scientists. And calcium chloride, you can see, I'm going to talk more about calcium chloride, but that tends to lower the pH even further. And the reason for that is how, I guess, when you add many of you probably know about cation exchange, and when you add a cation to a soil solution, that can induce various reactions, both in the solution itself, but also on the soil surfaces. So electrolytes have an influence on the cation exchange complex and the surfaces. So the history of electrolytes is interesting. When I was going back looking at those early papers, in that early paper by Sharp and Hoagland, they recognised or they used electrolytes even back that long. So they've been around a long time, and they used different electrolytes, and what they found was found to increase the hydrogen ion concentration or decrease the pH, which, as I'll describe, is a common effect of electrolytes. So that was recognised over 100 years ago. It was probably the most well-known work, and probably became a bit of the benchmark for the use of electrolytes in soils, was done by Schofield and Taylor at Rothamsted Research Site. And they essentially settled on a 0.01 molar calcium chloride that essentially, when they added it to a soil, they got very stable pH readings. They were able to, I guess, create that. Once they've added that, the soil salinity is within 2 or 3% of that value. So you'll have a little bit of salt in the soil in a non-saline soil. So what they're saying is, look, with that and I think it was a 1 to 2 ratio in that, they got very close to that value. So then I guess what they inferred from that was that they were able to reduce seasonal variation in pH. So if they were able to normalize changes in salinity in the soil, and I'll talk more about this on the next slide, that can remove seasonal variation in pH. There was also talk about stabilizing electrode readings. Electrodes don't work very well in very low ionic strength soil solutions or any solution. So they used electrolytes to try and stabilize the pH. And also to flocculate soils. So having a lot of soil particles around your electrodes can lead to issues, which are called suspension effects. And subsequently, people have used various justifications along these lines. So pH does vary seasonally in soils. So this plot here shows you seasonal variability of a soil. This is in the US. And these are 4 different measurement methods here. So you can see between May and September that we're doing 5 measurements. This is a field moist, so just measuring, putting the electrode in the moist soil out in the field. This is when they air dry the soil. So you can see the pH decreasing. So when they use calcium chloride, 0.01 molar, which is the standard calcium chloride extract even today, and 1 molar KCl, which is also used today. So, there's a few things in there. You can see how using electrolytes has changed the pH quite markedly from the field. First of all, you can see, particularly these stronger electrolytes, that they've decreased the pH over a unit or so. It has flattened out the seasonal variation. If you look at, say, this high ionic strength one, the variation across that year is much less. But I guess if and this is probably a question we discussed, should we attempt, if we know the seasonal pH variation is real and it has got all these effects on all these other parameters, should we just try and reduce it, average it out? Because that's essentially what you're doing. And does that remove some of our ability to use pH for predictive purposes? And then the second thing I'd say, in this case, we're not actually removing the seasonal variation. Like if this calcium chloride extract had removed all the seasonal variation, that should have flatlined that line. That should have flatlined at a pH value, because you had the ability to remove seasonal variation. It is not removing the seasonal variation. I think the way that people are interpreting it at present in many cases. But we don't, I think there's not a huge number of time series studies that we can probably explore this in more detail. And I guess, from my perspective, is this more the real number? This is what the pH was in the field before we dried the soils and before we added electrolytes. Should we be focusing more on measuring what it might be under those field conditions if we want to represent something more approaching what was actually occurring in the field? And I guess some of the seasonal variations we see in acid sulfate soils also relate to drying. Like we've talked more there about seasonal variations relating to salt. So this experiment just shows you within drying of an acid sulfate soil for 10 weeks, And you can see the pH here started at 6 and got down in this case to around 4. So these sort of changes do occur, big changes do occur, and you will not even out this sort of change with calcium chloride either. Which doesn't make sense to do that either. I had access, or it's online. There's a South Australian soils database, and I was interested in looking at how, with thousands of data points in South Australia, how those pH values between water and calcium chloride interact. And this is a plot of all the data where they're both pH and water and calcium chloride. That's a 1-to-1 line. So you can see in general that most pH water values are higher than calcium chloride, so they're sitting above the 1-to-1 line, but they're higher by a variable amount. So it's very difficult to predict what calcium chloride will do. It changes pH differently in different soils, and that probably is due to differences in the amount of hydrogen ions on the exchange sites and aluminium present. So it introduces an effect that on average is higher, but it's highly variable, and some pH values are lower. So it's not universal that it will always elevate pH. There are a series of data points here which are lower. And yeah, so the variability and unpredictability and lack of ability to represent seasonal variations is an issue in my opinion. And then I started to think, well, if I've got all this data, I'll look at ionic strength, and this 0.03 molar, which is 0.01 molar calcium chloride. The ionic strength is 0.03 molar, and that sits here. So many soils in South Australia, and this is the difference between the pH water, so subtracting that difference from pH water minus pH calcium chloride. So you can see in a lot of cases that's positive, so it's higher, but there's no clear relationship with ionic strength. But I would say that the assumptions that I mentioned earlier with use of electrolytes, that we will stabilize the ionic strength, get blown out of the water in South Australia there's so many ionic strengths above that. So what you're doing, yeah, you'll end up with, if you're diluting this with 0.3 with a little bit of 0.03 molar, you're going to end up sort of somewhere in the middle here. You're not going to get down to 0.03 molar, and these ones even closer to 0.03 molar, you're not going to be able to dilute to 0.03 molar. So this idea that we can maintain constant ionic strength in our soil measurement solutions, I think is a bit of a fallacy as far as I'm concerned. And then, and this is particularly an issue in variable salinity soils as well. There's issues with pH electrode calibrations with variable ionic strength. So as I mentioned earlier, in the definition of pH, they talk about defining it with relevance to a buffer solution. So that operational definition, and many of you will have done your pH buffer calibrations. Well, those buffers, most of those commercial buffers have a very high ionic strength, and the difference probably between those ionic strength buffers greater than 5 decisiemens and many non-saline soils is over 100-fold. So we've got a very big difference between what we're calibrating, the medium, the operational definition of pH, and then what we're measuring in those sort of soils. And it could be for the rarely saline soils, it's different. It's higher than the buffer values as well. So really, for accurate pH measurements, and this is sort of a well-established fact in all the ocean acidification measurements and the work that they've done there, is that you're really needing to be around the same ionic strength as the buffers. So to do that in the soil, you would have to make buffer solutions of a range of ionic strengths to match your soil ionic strengths, and you might then be able to have some confidence in the accuracy of your calibrations. At the moment, we often just calibrate in these commercial buffers and accept the values that they give, but it introduces another source of error. So I think, yeah, one aspect in the project we've been working on is spatial variability in pH. So if we've sort of got a typical agricultural paddock in South Australia, what do people think might be a typical range of pH values in, say, a couple of units, or 2 or 3? Yep. That's pretty good. Yeah, that's a good point. Probably more the surface at this stage, but I guess I will talk about the depth though. It's a good point. So I guess PIRSA has been out there mapping pH using these various machines that they tow, and they've got a platinum electrode that comes down to the soil at defined intervals. And a lot of the data I've seen is this is surface soil pH. It only goes down maybe a few centimeters. There's often a very large pH, 2 to 4 pH unit variability, and lots of data. And it's quite surprising to me how variable it is. And I think it's got some big implications for, if you go out into the middle of paddock and you just happen to choose this spot here and you say, well, this is a non-acidic soil, but if you come and happen to hit this spot here in your random sampling, and if you're only doing a few samples, there's a chance that you'll do that, and that's a pH of 4. So you've got a huge amount of variability, and when I think we're just beginning to appreciate that, and it's got some pretty big implications, I think, for agricultural management. And then, as Matt mentioned, what we see on the surface, like an iceberg, can sort of, surface of a soil can mask what's underneath. So this is a plot from Ruby Hume, one of PhD students from one of the PIRSA trial sites. And I haven't got the, this is from 3 different locations. I've got to put the legend on. But essentially what you can see is a similar pattern where at the surface, you've got quite neutral conditions. So if you were to go along and just grab a surface sample, you might say, these soils here are, fine for agriculture. But you can see here down at 10 to 15 centimeters, you're down below pH 5, you're down 4.5. There might be a lot of aluminium present. You'd expect a lot of aluminium to be present. And that could be a major constraint to your crop production. So there is some big subsoil issues in South Australia and elsewhere. And so there's a lot of effort being put into trying to manage subsoil acidity. And I guess, understanding how pH varies with time, with depth in the soil profile is really important if you're in an agricultural production setting. Because this might be where the roots hit and suddenly go sideways. They don't go any deeper. They don't like it in the zone. And that we do see that in a number of these trial sites where the roots don't penetrate through these acid throttles. And I guess, the other interesting one is in the rhizosphere. There's been some work. Petra's dad actually did some work, Petra Martian's dad, back in the day. This is a more recent paper from 2019 where they're studying pH in the rhizosphere using some, I think, really fine electrode techniques here and staining techniques. And you can see here some big differences between when you've got a nitrate fertilizer and ammonium fertilizer, and also when you've got a high calcium carbonate, you've got very limited influence in the rhizosphere in terms of it's buffering the effects of the roots, whereas in the lower calcium carbonate soil, you can see a much bigger effect within the rhizosphere. So I think there's a lot more work that we need to do, and probably in conjunction with our plant scientist friends as soil scientists to really probe rhizosphere pH in more detail and understand how plant processes are interacting with the soil and some of the, I guess, implications of that for nutrient availability and other factors. So I guess as part of my group, we've had several students working on what we probably call novel spectroscopic measurements of pH through different methods. And I'll briefly describe those. So Actually, Sorensen and the early pH measurements, they used indicator dyes, but there's been a lot of development in indicator dyes. In fact, for ocean acidification, most of the most accurate measurements down to the 3rd or 4th decimal place in pH are done now using indicator dyes, because they are much more precise, and some of those issues that we've talked about with electrodes don't occur with indicator dyes if you can use them, I guess, in a very, I guess, controlled way. So how they work, and this is some work that we've applied these indicator dye techniques to soils, is you can measure, this is the spectra, the UV-vis spectra of a soil extract, and this is when we've added an indicator dye. And this is what we call the acid form of the indicator, and this is the base form of the indicator. So the ratio of those two go up and down according to pH. And you can use basically the ratio of those two peaks along with knowledge of the equilibrium constant for the dye to calculate pH very accurately. If you know that equilibrium constant, you can calculate pH. And this is a paper where we measured pH with a spectrophotometric method with Seamer's PhD, and showed that, we could attain, very accurate measurements relative to the pH electrode. But as we've discussed, there's some also issues with the pH electrode, but certainly we can get, very, very accurate measurements using these indicator dyes in soil. And just as a bit of an aside, we've been using or developing these new indicator dyes for the PIRSA project, which have a much stronger color change around pH 5.5. So this is a site where there's a topsoil that's been limed, and you can see this purple, it's greater than 6, and you can see you're getting down below about 10 centimeters, you're going into this acid layer. So some of the dot or the indicator dyes many of you will have used, those ones you can buy at Bunnings, don't have a very precise color change around pH 5. So we're working with PIRSA and trying to, I guess, come up with a better dye for acid soils that is more useful, particularly in the extension setting, so you can show to farmers, look, you can see the effects of your lime here, and you can see where it's got to and things. And we've also been working more recently on a lot of infrared spectroscopy measurements of both carbonate and soil pH. So infrared spectroscopy is essentially a method where infrared light gets shined at the sample. Some of that light is reflected, and the wavelengths that aren't reflected, essentially things that have absorbed or light radiation that's absorbed by the soil matrix, and different components of the soil absorb different wavelengths of that infrared light. So this just shows you an example of a spectra here, and OC's organic carbon. So you can see some areas that this is corresponding to organic carbon. I think this is smectite clay, quartz, etc. So you can develop a bit of a signature of many soil components. One thing Ruby, my PhD student's been working on, is detecting carbonate, which is the key buffering component of pH in soil. So this just shows you the spectra of soil that she spiked with different concentrations of carbonate. And you think, oh, that looks pretty similar. But then when you zoom into these key regions here, you can see these big changes here. So this is where the carbonate is being detected in these wavelength regions there. So this is proving, I think, quite a useful method for understand soil pH buffering that we've got, and also liming of soils. And Ruby's also using this to predict pH, and we're getting pretty good results using mid-infrared. This shows you lab measured pH, and this is some predicted at higher resolution intervals using mid-infrared. And so we're picking up the ability of mid-infrared is, I guess, a little bit cheaper, and you can get more high resolution measurements potentially, is where we think we're trying to take the technology, which comes back to that ability to map pH and understand it maybe and measure it more often in the field. So we're working on that, and looking quite promising. R squareds of about 0.8. So infrared is not as accurate as wet chemistry, but the advantage is you can get a lot of measurements quickly, potentially. So some of the accuracy will offset the, some of the reduced accuracy is offset by that ability to take more samples. And this sort of was highlighted in study by my honours student, Bethany Sleep, where she mapped using visible NIR in the field and predicted pH. And this is sort of what she measured in the lab using traditional methods. And here's her map that she did with her infrared method. So while there's a few differences in places, generally she was able to pick up the patterns, and in an agronomic context, I think she mapped this in the field in a couple of hours. So she had data that was, you know, she collected infrared data in a couple of hours from that paddock. So potentially the advantage is you might be able to help the farmer redirect lime resources and target areas. So in this case, you'd say, look, this needs a lot more lime than down here. Maybe this wouldn't need any lime. So some of these techniques, that's probably where they fit into that precision agriculture space. And that's my last slide. But as a brief summary, pH was invented over 100 years ago. It has got some complexity of definition and measurement, as we talked about. I think it is a master variable in soils, but I think there are some measurement issues, misunderstandings, and really challenges to relating what we're measuring in the laboratory to what we might call the real pH in the field. Electrolyte solutions, in my mind, are particularly problematic, and they're not really doing what they set out to do in many soils or many contexts. We talked about that high horizontal and vertical variation in pH, which is often not being picked up, I think, in many surveys, and that even in the rhizosphere, obviously down to that really fine millimeter scale. Some of these new techniques are promising. It gets you around problems with electrode stability and calibration with variable ionic strength and things like that, that I think will be a way of the future, but we've got a, I guess, long way to go there. And yeah, but overall message is probably we just need to probably understand when we're undertaking pH measurements, what we're doing, what sort of soil it is, and check some of these assumptions that we might make about measurement conditions and things. And on that note, I'll sign off. Thank you.
Okay, thank you. It's great to hear some actual calls after a seminar rather than just.
Thank you.
So yeah, for those that are online, please enter questions into the chat, into the question and answer session. We brought that up online, so we'll answer those. But first, I'll hand over to the audience for questions.
Aaron, yeah, what? Yeah. That's a good question. So when you say relying on colour, so the spectroscopic measurements, yeah. So essentially what the method SEMA developed is centrifuge the soil solution. So you're removing the big particles by that method. In that slide that I showed you, it had the background spectra of the soil extract. Essentially that background spectra gets subtracted from the spectra when the dye is added. So the background absorbance levels are, I guess, corrected for. In saying that, yeah, it tends to be more in the UV range that you get the organic compounds absorbing, whereas these dyes are pushing more on the UV vis. So hasn't been a problem to date, but we probably didn't work with really organosols or any really hugely peaty or materials and things. So I think it's an area that is something that could be an issue if you had a really strong. But in theory, we can correct for it quite accurately by running the spectra before you add the dye and then after you add the dye. Yeah, that's what we did. Yeah. Sorry, your question up the back there, was it? Yeah. Yeah. Thank you. Yeah. They are, yeah, no, it was a fairly routine spectrophotometer. I think it might have been yours, Annie, back in the day. The one thing we did do was add a water bath and a temperature control onto the cell. The dye, for the most accurate work, the dye is also affected by temperature. So for the research work, it's a relatively minor effect, but we control temperature in the cell where the cuvette is. So that was the only customization we did to a standard spectrophotometer. The other thing we have tried that I didn't go into is reflectance probes and spectrophotometer we could take to the field. Like there's a lot of portable spectrometry solutions now you can take to the field that are battery powered. They're getting pretty good, I think, stability and control now. They're not as accurate, I think, as the lab spectrophotometers, but they're probably, accurate enough for the sort of stuff we're doing. So yeah, it is pretty standard equipment. Yep.
We'll take one from the floor, one
yep.
online and then
yeah.
So, Luke, you put a lot of doubt in my mind about the validity of soil measurements. Certainly, what we might get from the labs, diagnostics. So Georgios has a question here. So about when getting a lab report for soil pH.
Yeah.
How confident can we be in the value that we do go to labs, give any indication on precision?
Yeah, look, I think you can be confident if it's an accredited laboratory that you will get a result that is, certainly accurate in terms of they have, they've calibrated their electrodes. They have undertaken quality control and various, I guess, processes to make sure that you're getting, so you can probably get a reproducible number. As I described, whether how you relate that then back to the field is probably where I have the most, some of the most issues, because I presented those plots where it shows very different field pHs and so you probably just need to be careful, and obviously if you're getting calcium chlorides, understand that that's relating to a system that's been perturbed. Probably, I didn't really talk about it, but probably for soil acidity, where you're adding calcium carbonate into the soil is probably one of the more, I guess, I don't think it's necessarily bad calcium chloride measuring pH, because you add all this calcium in anyway as calcium carbonate to try and neutralize the soil. So in a way, you're getting a worse case when you've bumped a lot of calcium in. So if you're doing soil acidity stuff, liming requirements, I think there's less worry perhaps. So you can be confident in the lab's ability to measure a number reproducibly, but yeah, just be cautious about how you interpret that. Yep.
So in terms of trials.
Yep.
Trials. So you show the map
Yep.
of soil pH, for instance.
Yep.
And then you've got wheat plants, whatever, growing
Yep.
on top of that.
Yep.
In terms of what the plant actually sees and how it responds, what
Yeah.
variables do you get there? Trials. So I guess
Yeah.
is what the plant can cope with and
Yep.
how it buffers pH itself.
Yeah. No, it's a really good question. I mean, at the moment, I don't think we have a really clear idea what's really going on in the rhizosphere. We're not doing any of this sort of, that plot that we'll go back. So I think that's probably an area that working with some of you plant scientists, is to try and, try and develop maybe some of these techniques a bit more here at Waite, where we can can look more closely at the rhizosphere, but typically, yeah, that is not subsampled, and I guess it has some challenges when you're talking millimeter scale to subsample, and that's maybe where these indicator dyes can really come in a bit more and start to probe that. But to do it in the field, still challenging, I think. Yeah. Tim, yep. Don't worry, I have as well. Yeah. I'm not blaming anyone. Yeah. Judging anyone. Yeah. Yep. 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 Yeah. Yep. Yeah. So Tim's asked, I guess, about using more high throughput methods that I guess you use them a lot for nutrients, do your colorimetric analysis, and whether there's possibility, I guess, of applying these indicator dye techniques in that. And I think, yeah, that definitely, if you can measure the right wavelength, so those dyes that I mentioned, where do we do that? Sorry. Yeah. So essentially you have to be able to measure at those peaks. That's all you have to measure. So as long as you can measure the UV vis spectrum and pull out numbers at those peaks, then it's a ratio of those peaks that are important. So yeah, it would be interesting, I think, to try and look at that. In terms of, Tim also asked about EC, and EC is typically measured by electrical conductivity. I don't know that there's any dye methods that could do that. Redox sensitive dyes, you can do redox.
You can get specific ones.
Yeah, can you? Yep. Yeah, that's right. You could get a chloride one or something like that would be probably as a proxy for EC. Could you? Or, yeah. Yep.
Ultimately, probably what you need is a nice GM plant that can tell you all of this.
Yeah.
That's what's most important. It's what the plant senses.
Yeah, and that's where, you know, maybe we just need to work with plant scientists a bit more. And, you know, this is really interesting. We've got a lot of calcium carbonate soils in South Australia, so we're probably a lot of the time in this sort of situation. But obviously plants are influencing, yeah, proton flux, and I don't really understand that aspect, and that's why I need to sort of probably work with, yeah, more plant colleagues in this area and try and unpack it.
So Mike has a question, Mike.
Yeah, Mike, thanks, Mike. Mike's asked about mid-infrared and have we looked at MIR to measure soil pH itself? Yeah. So this data here, Mike, yeah, I think this was the data. So this is mid-infrared. I haven't put the calibration curve up, but it's around that R squared of 0.8. And some of the work actually that Les Janik and I know Mike, you are probably involved in. It's a little bit of an interesting one, pH, but it's not like carbonate where there's a mineral that is directly absorbing infrared. It's more of a, I think the spectra is a proxy for pH. So we know pH in soil is influenced a lot by organic matter and clay content and some of these others. So organic matter and clay are very readily, you know, measured by infrared. So I think it's sort of like a secondary proxy measure of pH. So we do have to be cautious, and I think you do have to develop calibrations, probably, you know, to have confidence in using it. But certainly there's several studies in the literature showing around that accuracy of 0.8, and which, you know, is I think showing that it has promise, at least for that agricultural mapping, or maybe deciding where you're going to sample. Have you got a paddock with lots of variation and you need to think more carefully about your sampling strategy? So Yeah, we have, Mike. Ruby's got a paper submitted on that at the moment. This is some of the plots out of it. Thanks, Ruby. Yep.
2 hours to sample using the
Infra- Yeah, in the field. Yeah. Yeah. Well, I think, yeah, days basically. You bring it back, you're drying it and calibrating. There are some, issues with infrared in the field, particularly mid-infrared. Moisture content is one that can interfere. So, yeah, but I guess we are working on some of that field applicability of infrared techniques. We've got a project with the engineers for soil carbon with an autonomous tractor and soil sensing. So we're trying to work on it. How portable, making these mid-infrared, and Les Janik is involved too. Pioneered a lot of these infrared methods here at Waite, actually, which is very valuable to have him around. Yep. Yep. Yeah. 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 All right. Thank you.
Or maybe you want
to. Yep. Yep. Yeah. Well, that's the question I sort of pose, I guess. These are just plots of the pH itself. I guess what worries me a little bit is we go and do the calcium chloride to try and even out pH variation, and we say, well, these are the values, but the real pHs are much higher. Are we sort of, I guess missing something in terms of some of those relationships, or ability to predict some of those relationships with nutrients, and because of our attempt to alter the pH and even it out, even if it's unsuccessful in this case, to even out seasonable variability. So I can't really say, but it would be an interesting area. People have talked more about salinity variation. So as you get, obviously summer and the salts are concentrating in the unsaturated zone of the soil profile, that is sort of what people are talking about as driving a lot of these seasonal variations. I think possibly oxidation reduction, maybe if the soil is quite wet as well. It's my feeling. You're a plant breeder, yep. 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 Yeah.
I think we'll have that on we'll close up the seminar, carry on that
Conversation.
Thank you very much, Luke, for
yeah.
giving us your seminar today.
No.
Thanks to the audience for
Thank you. Thank you. Why is that? Stop sharing.